Hello, my name is Mariah Hoy, and on behalf of the NINDS Office of Research Quality, today I'm going to be presenting on how you can maximize data transparency using Rigor icons. I want you to imagine that you're at a scientific conference and you see an oral or poster presentation, and the speaker is showing you a bunch of results, but there's really little to no context regarding how those experiments were conducted and how the results were obtained. You may find yourself wondering, how many animals were used? How many cells per animal? Were experimenters blinded and masked? Were the groups randomized? What statistical test was used? But why do we really need this information? Well, philosophers of science, such as Francis Bacon, recognized that the scientific method could be compromised by human nature, stating in his book, The New Organon, once a man's understanding has settled on something, it draws everything else also to support and agree with it. And further, it is an innate and constant mistake in the human understanding to be much more moved and excited by affirmatives than by negatives. And we know this as unconscious bias. And unconscious biases can definitely be at play when we are conducting our experiments. So how do we address these unconscious biases? Well, in this 2005 review, David Rantahoff proposed the reliability of a study is determined by the investigator's choices about critical details of research design and conduct. So in other words, it's really our responsibility to make sure we conduct our experiments in the most rigorous way possible. But he does say that bias is unintentional and unconscious. So how do we reconcile these two? He provides a simple solution. The process of addressing bias involves making everything equal during the design, conduct, and interpretation of a study, and reporting those steps in an explicit and transparent way. So we have to make everything equal between comparison groups, except the one parameter we are testing. So how do we do this? For one, we can conceal the identity of comparison groups by blinding and masking experimenters during data collection and analysis. This practice helps prevent experimenters from unconsciously treating samples differently based on the identity of the sample. We can also randomly assign samples to experimental groups using a random number generator, for example, to help ensure unconscious biases are not contributing to group allocation. We can also predetermine appropriate sample sizes to ensure experiments are adequately powered to detect a difference if it's there. And this also helps prevent things like N-hacking and P-hacking, or just continually adding samples until significant results are obtained, as this can increase the risk of false positives and incorrect inferences. Finally, we can predefine criteria for excluding samples or outliers, which can help prevent experimenters from unconsciously justifying removals of samples that may actually represent the true variance within the population being sampled. So what happens when we don't employ these practices? Well, unconscious bias seeps in and can skew our results. This systematic review and meta-analysis assessed 29 studies of a drug that was tested in animal models of stroke and found that there was minimal effect of the drug in these studies where they reported and presumably used these various measures to minimize the risk of unconscious bias, such as blinding and masking, randomization, and sample size estimation. So you can see that the studies with higher reporting quality actually found little to no effect, whereas the samples that didn't report these measures found a large effect of the drug. So again, as Francis Bacon said, we are much more moved and excited by affirmatives. So if you don't take measures to minimize bias or report them, your results might be skewed to look better than they actually are, and all because of unconscious bias. So the bottom line is we need transparency. Without transparently reporting research methodologies, we can't really assess the quality of the research. Now, as a presenter, you might say, hey, but wait, I did all of those things. I just didn't have the time or the space to show it. Don't worry, we've got your back. Introducing time and space-saving Rigor icons. Rigor icons are small symbols that can convey those important experimental design details, like blinding and masking, randomization, data exclusion, sample size estimation, whether an experiment was exploratory, and then whether the data are accessible in an online repository. And all of these icons are freely available on our website. Maximizing data transparency is easy. Take this figure with bar graphs and ill-defined error bars. You can't see the individual data points, you don't know what the sample size is, and you have no idea whether the experiment was conducted rigorously. The same data are represented on the right as a quantile box plot showing the mean by this orange diamond, the sample size, the individual data points, and the range for data spread. And now we've added these Rigor icons to indicate that the experimenter was blinded and masked, that samples were randomly allocated to groups, and that sample size estimation was performed in advance. As you can see, this graph on the right, with virtually the same amount of space, conveys much more detailed information about both the methods and the results. Another use of Rigor icons is they can be used to help distinguish between exploratory and confirmatory experiments. So let's say you conduct an exploratory experiment where mice received some perturbation. You can add the exploratory icon to indicate that this was a hypothesis-generating experiment, and you can also explicitly show the sample size, which increases transparency. In this example, single-cell RNA sequencing was performed, but one of the samples did not meet the minimum threshold of live cells that was one of your inclusion and exclusion criteria, and so this sample was excluded from the analysis, as is depicted by the data exclusions icon. Now, in this exploratory analysis, you identified a subset of cells with a unique response to the perturbation. So cool. And so now you want to follow up on those exploratory findings by performing a confirmatory experiment, in which you will likely be much more rigorous and take additional measures to minimize unconscious biases, again, such as blinding and masking, randomization, sample size estimation, and predefined inclusion and exclusion criteria. Here, I'm showing the experimental schema where Rigor icons have been added at the appropriate steps. You performed a power calculation and determined that the sample size of 12 mice per group was needed in order to be adequately powered to see a meaningful difference between groups for your behavioral task. You randomized samples to experimental groups, and you blinded or masked experimenters during the experiment as well as the analysis. 3 days later, you perform this behavioral task, and then further, you randomize the mice that underwent that behavioral task, and 7 of those mice are going to be used for immunofluorescence, and 5 of them are going to be used for Western blotting. As another example, here I'm showing the exact same experimental design schema, but now instead of blinding experimenters at the very beginning of the experiment, you perform blinding later on for both the behavioral experiment in the observation and the analysis phases, as well as during immunofluorescence for tissue prep, imaging, as well as analysis. Blinding, however, was not performed during the Western blotting experiment. And again, this is just a way that you can use schema to indicate at which step practices occurred and be more transparent. Finally, it's really important to add all applicable icons to each result panel of a figure, since not all panels may have followed the same experimental design and or been collected and analyzed in the same way. So for instance, in this analysis, experimenters were blinded and masked, sample size estimation was performed, and the data are available in an online repository. But for the shoal analysis, one of your samples actually met your predefined exclusion criteria. And so that sample was removed. And so you're depicting that with the data exclusion icon. And you're also showing that this sample was removed from the perturbed data set. So wow your audience with transparency and start using Rigor icons today. I've included the QR code as well as the link to the webpage, where you'll find a flyer explaining the use of the Rigor icons, the individual PNG files for all of the Rigor icons, as well as a PowerPoint with a table of the icons that can be used as a key. Thank you.